Hello, this is DNA replication with Jake. So this is DNA. You ready? Anyway, so DNA is a type of macromolecule, one of the four. It is needed to carry, it is the genetic information for all living things. Its monomer of the macromolecule is a nucleotide. A nucleotide is made of a sugar, a phosphate, and a nitrogenous base, one of four nitrogenous bases, including adenine, guanine, thymine, and cytosine. And on that sugar, in the nucleotide, on the sugar, it's a 5-carbon sugar, and it's numbered from the top of the peak. It's like a pentagon. It's numbered from the top, 1, 2, 3, 4, all the way to 5. And that's important later. Anyway, so I'll come back to that later. So DNA replication, how it needs to replicate to make new cells and, to make haploid cells to carry on genetic information and to, when other cells die to make new cells and all that good stuff. That's why you need DNA replication. And it happens from old DNA making new DNA, and that's called semi-conservative theory? Conservation. Semi-conservative replication is the correct term for that. And how that works is that it splits and unwinds into two. I'll explain how that happens later. And that each strand makes a new one, and eventually it makes two new strands made of one old and one new times 2. Sweet. So how that works is that DNA helicase starts at the point of origin, which is correctly called a origin of replication. In the prokaryotes, there is one origin replication because it is circular. So it only starts one place and it goes all the way around. In eukaryotes, there is many origin replication. So at that origin replication, the helicase goes, it looks like a little Pac-Man. Look at that. It goes and it unwinds the genes. Like the joke, hey, I'm DNA helicase and I want to unwind your genes. Unzip your genes. Anyway, so it unzips the genes. It tears apart the hydrogen bonds that are in between the nitrogenous bases. And that is what helicase does. So once you get a good unwinding, that's how this became known right here. At the replication fork. This is replication fork. And that's where it unwinds from. Good. So now let's take a look at what we have here. That, as you can see, oh, by the way, it starts with the top box. That's a thymine, adenine from the fork, origin replication. And yeah, it just kind of starts there. Don't know why, just kind of does. And so, anyway, it starts there, tears apart the hydrogen bonds, and from there, oh, by the way, from the Chargaff's rule, A pairs with T and C pairs with G. So that's always good to know. And so they rip apart, and so as you can see, like A with T, T with A, and so the new strand will have to match the old strand exactly for DNA. Never mind. So for DNA, it matches it exactly. How that happens on one of the strands, the leading strand, this one, what it does is that the DNA polymerase 3 is what lays down the new strand. It goes in the 5' to 3' section. Now remember that sugar that we had before with the 1, 2, 3, 4, 5? It goes from the 3 corner to the 5 corner, and that's how it replicates. So that's why that's important. So this DNA polymerase goes, and it goes, here's the 5' section, Here's a 3 prime. It just goes, woo, it goes all the way, and it makes this bad boy right here. That A with T, C with G, all that good stuff. And that is the leading strand. All fun and games. Oh, that's the easy one. And oh, quick side note, in these, to relieve tension from the fork, back to that fork thing, because, you know, you're stretching these things and it's going to be, you know, tough to do. So while you're stretching those things, they're to relieve tension, kind of like how you're like stretching a rubber band in a circle. That's hard to do. To relieve the tension, there's these things called topoisomerase, these red things, and they just relieve the tension. That's about it. It's always fun. Anyway, let's put that on there. Good. Anyway, so we made the leading strand. Ah, yes, isn't that great? But by the way, what you need to start always is the RNA primer. That's just where DNA polymerase needs to start. That's how it goes. Because it goes here, because it needs a free 3 prime, yes, needs a free 3 prime section to start its making of new nitrogenous bases. 3 prime section? Yes, that's correct. 3 prime section. Anyway, leading strand, it just really needs 1, and that's about it. But lagging strand, that's where it gets fun, is that, so it can only go in the 5 prime to 3 prime section, and that the 5 prime section is here and the 3 prime section is there, but it wants to, you know, take care of this stuff down here before it takes care of this stuff. So how it works is it needs to do it in fragments. And what we call those fragments are the Okazaki fragments. We call that the Okazaki fragments. Those are these things right here. Okay, so it takes, what happens is there's an RNA primer that starts right here, then the DNA polymerase 3 synthesizes those things synthesizes it in this direction, to the next RNA primer. Sorry, this started right there. It went from this RNA primer to that RNA primer, and it synthesizes those right there. And then, so then this goes away, and then, you know, makes those ones and it makes these and yeah, awesome, right? Look, now we got this whole big thing, and it looks so pretty. But now we got these big RNA primers in the way, so we're gonna take these out, and in its place, DNA polymerase 1 is going to put down some nitrogenous bases in their place. And then, so it's going to lay down where the RNA primers were, pretend we're having some things there. Let's go lay those down. And that's its purpose, to replace the RNA primer's place. And then finally, the ligase, this little enzyme right here, is going to make the phosphodiester bonds between those to replace what the DNA polymerase 1 made with what DNA polymerase 3 made, not 2. And so now we're going to take a quick breather. Enjoy the picture here that we're making. That's a lot. Can we pause this video somehow? Anyway. So, it's a long breather. Here it is. I don't want to do this again. So, we're going to take his hand sheet right here. And it's going to be in the video because it's a lot of fun. Anyway, so, like when you were watching this, it did in fragments, and the correct term for that is discontinuous. It can't do it in one continuous thing like the leading strand. It has to do it in segments called discontinuous. Leading continuous, lagging is discontinuous. And then, like I said before, this is a double helix, just to reiterate that, just in case you didn't quite get that. And this outside that you see, this pink right here, is the phosphate sugar backbone. Because remember, on the monomer, the monomer, which is called the nucleotide, excuse me, the monomer, which is the nucleotide, has a sugar and the phosphate on the outside and then a nitrogenous base on the inside, towards the middle. And the correct term for the 5 to 3 primes, this was the 5 prime section to the 3 prime section, and this is going the opposite direction. The correct term for that was anti-parallel, that they go in opposite 5 to 3 prime ways. Oh gosh, excuse me. So, and just to throw this in there, adenine and guanine are purines, and cytosine and thymine, and if it's RNA uracil, is pyrimidines, and purines are double ringed and pyrimidines are single ring. That's just going to matter when they're attaching to the sugar, that if they have, it's just if they have 2 rings or 1 ring on that nitrogenous base. And almost done here. That so RNA primer, just in case I didn't say this before, was made by primase. That is the enzyme that made that right there. And lastly, SSBP means, it sure means something. I will tell you that, folks. It means something. Single-stranded binding protein. Yes, that is what that means. And that is going to be this red dot folded in half that I will put right here. Now, I'll be brutally honest with you. Don't quite know what it means, but it is attached to the outside sugar phosphate backbone, and it definitely is a single-stranded binding protein. I'm Jake Moser, and this was DNA Replication with Jake. Have a good night, folks.